This is a recorded lecture for Chapter 1 for Microbiology 230. And this is really the introduction to microbiology as we go through this. This will be the introduction to the history and some of the background of microbiology as we move forward. My name is Dr. McGraw, and we'll get going here. All right, so let's talk about history of microbiology. The science of microbiology was officially born, if you will, I hate to use that term, in 1674. And strangely enough, it was by a gentleman who sold cloth, say fabric merchant. And Antony von Leeuwenhoek is how it's pronounced, who lived from 1632 to 1723, needed a methodology to decide the quality of the linens he was buying. And linen, if you know or not, is really decided by the number of threads per square inch. And so he needed a method to look at that, because sometimes people would try to swindle other people with that. So he made a simple magnifying glass. And after he started looking at the linens, he started looking at other things. And he studied lake water and observed that there were live elements, live organisms in this water. And he called them amicals, if you will, animalcules. And then Robert Hooke came along. He also credited with discovery, described the microscopical mushroom, which commonly bread mold, in 1665. So these are the people just discovering that there are organisms smaller than you can see with your naked eye. Now, the importance of these microorganisms is microorganisms are the foundation for all of the earth. In fact, the breakdown and buildup is really based a lot on microorganisms. They've existed for well over 3.5 billion years, and plants, animals, and modern microorganisms are all evolved from the ancestral bacteria. So our life really depends on our activities. In fact, we depend on the bacteria in our colon even to survive. If our colon was sterile, for instance, we would die. Now, there was a dispute over a long dispute, which went well, believe it or not, into the late 1800s, early 1900s, and that was whether life arose spontaneously from non-living material, like magic, if you will. And that was called spontaneous generation. Now, there were people that believed it and people that didn't, but there were a lot more people that believed it because it was magic and it was the way it's always been. The people that didn't believe it were three individuals that kind of stand out. One is Francisco Redi, Louis Pasteur, and John Tyndall. And each of these three contributed to disproving the idea that things just grew out of nowhere without any other basis. Italian biologist and physician Francisco Redi demonstrated that worms on rotting meat came from eggs or flies that landed on the meat. That was in 1668. He placed the meat in 2 jars, covered one with gauze. The gauze prevented the flies from depositing the eggs, and so no eggs, no worms. No eggs, no worms. And so it took another 200 years before convincing anybody to disprove that spontaneous generation of microorganisms, even though that was a pretty good study. One reason is it got conflicting results between laboratories. Remember, in that day, before we used scientific scientific methodology, people could sort of skew the test, the experiment, if you will, to get the answer they wanted. Now, multiple contributions helped define the question, finally. And in 1749, John Needham demonstrated boiled broth still produced microorganisms. Well, of course they would, and part of that is because the boiling it doesn't really eliminate all the bacteria in the broth. In 1776, Father Spallanzani, he contradicted Needham's results. He said boiled broths longer. He sealed the flask by melting the necks that are closed. The broth remained sterile unless the neck was cracked. So he said, well, if we put it in a closed system, then they will not have any growth. Now, even though that all occurred, controversy still was unresolved. Some argued he destroyed the vital force, whatever that is, necessary for spontaneous generation, the vital force magic, if you will. And then the French chemist Louis Pasteur came along, Louis Pasteur, and he's considered the father of modern microbiology. He's demonstrated air is filled with microorganisms, and he filtered air through a cotton plug and he observed the trapped organisms. So many looked identical to those found in the broths. So he made sort of a hypothesis that these are the organisms that were causing growth in the broth. So he developed what's called a swan-necked flask, and you see here it has kind of this little S-shaped neck, the flask, and he boiled the infusions, remained sterile despite open to the air. So the organisms couldn't get in there. They could settle here, if you were, but they couldn't get in. And so what he did is he showed that if he laid it down, the organisms could aggregate at the opening of the flask where the broth was at, and that it would start to infect that. So he said that the broth was sterilized, but air escaped, but the organisms can't get in. And the broth stayed sterile indefinitely. However, if you laid the flask on its side, what happened is the bacteria were able to get the broth. And so the biogenesis described the production of living things from other living things, which helped to disprove that spontaneous generation, which, by the way, was still under dispute even then. So some scientists remained skeptical. Pasteur's results were not fully reproducible, and the English physicist John Tyndall finally explained conflicting data. He proved that Pasteur was correct. The sterilizing broths required different times. Some sterilized in 5 minutes, others not despite 5 hours of sterilizing. So realized that broths made from hay contained heat-resistant microbes. The labs with contaminations used broth made from hay, and that's what happened is they used some organisms, you can't boil them away. If they're in a cystic form, then the cysts are able to resist some pretty high temperatures, and they can resist that heat. In the same year, 1876, German botanist Ferdinand Kohn discovered endospores, which is what we're talking about, and they're heat-resistant forms of bacteria. And that extreme heat resistance to these endospores explain the differences between Pasteur's results and everybody else who was trying to dispute him. Pasteur's broths made with sugar or yeast extract, and it highlights the importance of reproducing all the conditions as closely as possible when conducting research, so you don't change the factors in the research from one test to another. Use the same factors. Now we talk about the golden age of microbiology. As spontaneous generation was disproved, the golden age of microbiology was born. We discovered there were organisms, and they didn't didn't arrive from nowhere. They didn't arrive by magic. That truly they reproduced and they were available in the air. The principle that microorganisms cause diseases is known as the germ theory of disease. And most pathogenic bacteria were identified somewhere between 1875 and 1918. Now, they had no idea what viruses were in those days. We started work on what we termed viruses, but we didn't know what they were because we couldn't see them. You can't see a virus with a light microscope. And so understanding that microscopic agents could cause disease led to control efforts and huge improvements in the past century in human health, antibiotics to treat infectious disease, vaccines to prevent diseases, public health to prevent people from contaminating their own water supply. In fact, it's interesting because there are county agents in every county in the United States, and those county agents are part of public health. And they were developed when there was a cholera outbreak, an epidemic, if you will, throughout the United States. And most of the causes of that cholera epidemic were people were putting their outhouses next to their water supply. So the outhouses sewage would seep into the water supply, and they would self-contaminate. And so basically the first goal of these county agents was to go out in these rural areas, these farms, and have people move their outhouses out a distance from the water so as not to contaminate themselves and cause them to get cholera. Now, historical events in microbiology, some serious milestones occurred in microbiology in relation to other historical events, and you see this nice little loop here as you go through, and I'll let you look at that on your own. I won't go through that, belabor that, but it's really a very nicely detailed loop about what happens in the microbiology sense and what's happening in the rest of the world. Now, when I talked about scientific method, that's the part that we use now, or the method we use now. That's really how we go about running an experiment. We don't skew the experiment with our own biases. We try to make an experiment that has no biases. So the goal is you make an observation about something happening, and then you develop a testable explanation called a hypothesis. And you design experiments to test the hypothesis. In fact, in most cases, you design experiments to disprove the hypothesis. I know it sounds funny. It sounds negative, but in scientific methodology, we actually try to disprove the hypothesis we just created. So we look for ways to break it down. So it removes our biases. So if you say, okay, I believe this is true, and you have an experiment that proves it's true, it may contain a bias to prove it's true. Or if you believe something to be happening, and your hypothesis is to prove that it didn't happen, then you're not really showing your own biases in the experiment. So you do the experiment, you collect and analyze the data, and you draw a conclusion. Is this hypothesis correct or not? If the hypothesis is correct, then you move on and develop a scientific theory that says, okay, this is why it happens. But there's more than just one experiment. Lots of experiments have to occur to validate that your first experiment was correct. Now, if the hypothesis was not true, then you go back and you redo your observation, saying, well, that's not a true hypothesis, and you identify another hypothesis, something maybe that's closer to the truth, and you do the whole experiment again. So you communicate the methods and results and conclusions to other scientists. They redo your experiment to see if it's validated or disprove it. So scientific theory is an explanation supported by a large amount of evidence, not somebody's personal bias. Now, microbiology, look at a human perspective, as I said, we can't survive without microorganisms. There are bacteria in our gut that help us develop vitamins that we use to live on. Some examples of benefits are nitrogen fixation, they produce oxygen, there's degradation of materials like cellulose, also sewage and wastewater. So these microorganisms break down the byproducts, the after-products of animal feces such, and they help to make it so it's actually useful to the environment. But microorganisms have also killed more people than ever killed in a war. They've been used as weapons and could be used in bioterrorism attack. If you remember the days of September 11th, if you were alive then, that basically what was happening is they used anthrax as a bioterrorism weapon. Now, the interesting thing about that anthrax that was used, that was sent to the Congress and all that, guess what? They went back and did the genotype of that and found out where it came from originally. It came from our own Centers for Disease Control, CDC, in the United States. What had happened is the CDC keeps all these cultures of all the bacteria viable, so you just can study them. Anyway, and what happened is that they had loaned out a sample of that to somebody, and they never came to light who that was, and they would loan out that sample, and that sample was used to create bioterrorism weapons. Now, all surfaces of the human body are populated by microorganisms. That's simple as it gets. Your skin actually has staph epidermidis on it, and the staph epidermidis has a low pathogenic value, if you will, but what it does is it tends to eat all the resources that are available on your skin, so that other more infected bacteria, more pathogenic bacteria, cannot cause the disease. It's a way to sort of use up the food, as it were. Now, beneficial microbes are termed normal microbiota or normal flora. They prevent diseases from competing with pathogens. With competing pathogens, and they help develop an immune system response, and they also aid in our digestion. Now, pathogens, by definition, damage body tissues. They cause disease symptoms. They have a negative effect on the body. Where these microbiota or normal flora have no effect on the body. We live in a symbiotic relationship. Now, microorganisms in the environment, they help recycle of our nutrients. The oxygen production through photosynthesis helps to add more oxygen to the air. They produce nitrogen fixation, which is helpful to our development and also to development of our food. And they help to decompose their material. For instance, cellulose is degraded in the environment, and the digestive tracts are ruminants, if you will. And some of those include cows and animals that help to break that down. Now, application of microbiology include fruit production. If you've ever baked bread by hand, use yeast to make the bread rise, and Egyptian bakers as early as 2100 BC found that out. Fermentation of grains produced beer. Egyptians also, the tombs reveal that early as 1500 BC that they learned how to make beer. And we also have fermentation of milk to develop yogurt, cheeses, and buttermilk. It helps also in biodegradation, breaking things down. We degrade PCBs, DDT, trichloroethylene, and others to help clean up oil spills. We use bacteria to do that. And bioremediation, using microorganisms to hasten the decay of pollutants. So we use these microorganisms in our everyday life, if you will. Now, applications of microbiology include bacteria synthesize commercially viable products. They include cellulose for stereo headsets, hydroxybutyric acid, which is a manufacturer of disposable diapers and plastics. Ethanol is a biofuel. Hydrogen gas is possible biofuel. Oil, possible biofuel. Insect toxins like insecticides. Antibiotics for the treatment of disease. And of course, amino acids as dietary supplements. They also, bacteria also have been trained to produce other things, like in the case of insulin that people take with diabetes. At one time, insulin was extracted from the stomachs of pigs. What happened is when the pigs were slaughtered, they would take the pig stomachs, pig pork bellies, if you will, and they would extract the insulin from there, and they would purify it, and that would be the insulin they would create. Lots of problems with that. Sometimes the strength of the insulin you got was different from one group to the other. Sometimes people were allergic to the insulin because it was from another species, pigs, if you will. And so what happened several decades ago is they were able to train E. coli. The E. coli live in our gut, as a matter of fact, to produce insulin. They went in through recorrecting their DNA, if you will, or changing their DNA, such that they were able to produce an insulin that was pharmaceutically exact or pure, equal to the insulin that humans make. And it's called humulin. So if you have been around diabetics or you take insulin, then that's what they use now, is this humulin. So they've done a lot more than just these few things here. We also use it in biotechnology, use some microbiological and biochemical techniques to solve practical problems. Genetic reengineering, like I talked about for E. coli, and helps to introduce the genes into another organism. We can use it for to create disease-resistant plants. Insulin, like I talked about, production medication. So we've used them to our own benefit. Now, the microorganism is a model organism. There's wonderful model organisms. Metabolism, genetics are same as the higher forms. All cells are composed of the same elements. They synthesize structures in similar ways. They replicate DNA. They degrade foods via this metabolic pathway they have. And what is true of elephants is also true of bacteria. And that was by Dr. Jacques Monod, if you will, who said, what is true of elephants is true of bacteria, and bacteria are much easier to study. So what he's basically saying is that we use bacteria as a way to study our own humanity. Now, in medical microbiology, most organisms are not harmful. The bulk of them are not. And some are pathogens. And those pathogens tend to be organisms that live at our body temperature. They cause disease. For instance, influenza, which is a virus, by the way, caused influenza in 1918, 1990, called the Spanish flu. It killed more Americans that died in World War II, World War I and II, Korea, Vietnam, and Iraq wars combined. So more people died in the United States than with many of those wars. Modern sanitation, vaccination, and effective antimicrobial treatments have reduced instances of the worst diseases. And truthfully, we're using vaccinations now to even prevent cancer. If you think back, there was a big it was in the news quite a bit about 10, 15 years ago, where they developed Gardasil. That was developed at the University of New Mexico. I was happy to be there at the time. I wasn't part of the study, but I was an administrator then. And they developed the vaccine that we use now to prevent people from getting HPV. HPV is now part of the childhood immunization regimen. And what HPV does is we thought it used to cause only what are called sexually transmitted venereal warts. And unfortunately, what happened also is they started to create a change in the tissue, and it would develop into cancer. And it caused cancer of the covering of the cervix, and if you will. So we call it cancer in situ. And cancer in situ means that this would cause such a cancer that sometimes we would have to have their uteruses removed. Sometimes they'd have to have these extremely painful treatments, like they'd wash the top of the cervix with acid, all sorts of terrible things. And the idea was to get rid of that cancer. But now with this HPV vaccination, we were able to eliminate up to 20,000 women from having have to go through that in their lifetime here in the United States. Now, past triumphs, for instance, the smallpox was once a leading killer. In fact, smallpox was the leading killer since man ever annotated that people died from diseases. 10 million deaths over 4,000 years is pretty significant. Devastating or unspoken populations like the Aztecs in the New World. It was used as a bio weapon. Many Native American tribes were given these blankets which had smallpox in them, and so they would kill them basically. And that's one of the first uses of bioterrorism. Worldwide eradication attempts eliminated the disease, and so there's been no reported cases since 1977. Now we still keep smallpox growing in the CDC, and we still have smallpox vaccinations available. And the reason is because it's fear that somebody could use this again as a bioterrorist, weapon, and they could, because people no longer get the vaccinations. Plague was another major killer, and that was, if you will, Yersinia pestis. 1/3 of the population of Europe, or 25 million individuals, died between 1346 and 1350. At one time, because of all the wars and the Crusades and the plague, there are only about 3 million people in the whole world. Think about it, 3 million people in the whole world. Now, I'm from Wyoming, and I don't think there's 3 million people in Wyoming, to be honest with you. So imagine what that meant worldwide. And today, fewer than 100 people die of the plague worldwide. but it's still available. It still happens. We have a case or two, some 3 or 4 in the United States. And if you could, the reason is because there's we'll learn later, but there's a flea that plagues the rodent population and it harbors a bacterium. And that flea bites humans, and lo and behold, they get the disease. Now we have antibiotics that now can fight Yersinia pestis, which is the bacterium that causes the plague. Now, despite impressive progress, there's lots of work that remains. We really haven't delved very deeply into the viral diseases or diseases associated with poverty. Respiratory infections, diarrheal diseases cause most illnesses and death in the world today. In the United States, 750 million infections occur, and we still have about 200,000 deaths based on these diseases. And so it costs us tens of billions of dollars to support patients who get these diseases, where we could use a vaccine, which may cost a few $100 or maybe not even $100, and we could eliminate this kind of death and destruction. The emergent diseases continue to rise. You see E. coli. There are different kinds of strains of E. coli all through the United States. We had E. coli outbreaks in 1982. AIDS says 1981 here, but interestingly enough, we know it existed in the 1960s. Legionnaires' disease, which is a bacteria that occurred in the American Legion Hall. Swine flu, which is 2000 here. Actually 2009 in Mexico. We had in the United States in 1977-78. And then cryptosporidiosis, if you will, in the United States, down in the southeastern part. You can see as we go through all the different diseases. There's Ebola, hepatitis C, which is becoming a big deal for elders now, hepatitis D in Italy. You're starting to see human and equine morbillious, if you will, in Australia. And you start to see all the different kinds of flu we have, and they're outcropping. Emergent diseases mostly are newly recognized, but there's multiple examples. The swine flu in the United States in 1977, severe acute respiratory syndrome, SARS, multi-drug resistant tuberculosis, Lyme disease, hepatitis C, acquired immune deficiency syndrome, AIDS, if you will, hemolytic uremic syndrome caused by E. coli, the hantavirus pulmonary syndrome. And we tend to see those in the Southwest. They're actually caused by what's called a deer mouse, and it's from the feces of the deer mouse. Mad cow disease, which is the encephalopathy that starts to leave, sort of change your brain into Swiss cheese in some ways. And then West Nile virus, and finally Ebola virus. Now, I put the Ebola virus here in red because it's kind of important. They're in the middle of developing a vaccine for Ebola virus that's 100% effective. They're just going through the paces now, but everything about the Ebola virus is starting to show to be very important and very effective, which means that the countries, especially in Africa and those areas where Ebola is very common, they can vaccinate those populations and basically make Ebola virus go away, which is pretty important. Now, changing lifestyles increases opportunities to spread the disease. For instance, closer contact with animals like hantavirus, evolution of infectious agents previously unable to infect humans like HIV and AIDS, which was originally affected monkeys, SARS and Ebola viruses, which were other animals. And we're having re-emerging diseases because vaccination can become the victim of its own success. We see that. People don't have the first-hand knowledge of dangerous diseases, and it leads people to fear vaccines more than disease. It's really interesting that there's a big push that vaccines cause autism. And just to let you know, there have been 26 studies, not related to pharmaceutical companies, by the way, but public health studies, done in 13 countries of over 100,000 children to associate, to try to figure out if any vaccinations cause autism. Now, these are different countries, so their childhood schedule for vaccinations are a little different from country to country. So they checked all these countries, did all these studies, and they found not one single incidence, not one, that was associated with a vaccine causing autism. Interestingly enough, there have been several studies that show that long-term use of video games can cause autism. It overpowers the nervous system and can cause autism. But nobody's standing out in front of the video game store and saying, no, don't sell the video games, or, if you will. So it's kind of interesting how it all works out. But just so you know, 13 countries, 26 studies now. It used to be 25, 26 studies, and over 100,000 children. No association. Oh, by the way, when I'm thinking about it, there is one. Read one study that was done, which has been disproved, by the way, which was done in the United States, which said the autism was caused by revaccination. That study has been disproven. In fact, even the person that put out that study came back and said he had skewed the data and it wasn't real. So, emergent diseases, pathogens can become resistant to antimicrobial medications like tuberculosis, which used to be very much easier to treat, malaria, and C. difficile, Clostridium difficile, which is it can be a very powerful bacterium which can harm us, but usually it's in such low quantity in our colon, nothing happens. But if you put people on long-term antibiotics, the antibiotics kill off all the larger bacteria, the larger groups of bacteria, and allow the C. difficile to create what's called intermembranous colitis in our colon. We have increased travel immigration, so many diseases that are eliminated from developing countries still exist in many parts of the world, like malaria, cholera, plague, yellow fever. There's a change in the population. We have weakened immune systems, the elderly, and people with HIV and AIDS. So chronic diseases can be caused by bacteria. For instance, peptic ulcers can be caused by Helicobacter pylori, H. pylori. And possible indigestion, Crohn's disease, others could make us more susceptible to certain diseases. Now, the second golden age in microbiology is what we're talking about now, and that's where we really started to get into breaking down bacteria and looking at them not under a microscope, but looking at their genotypes. That means there's a phenotype and genotype. Phenotype are the characteristics that an organism has that you can see. Blue eyes versus brown eyes versus green eyes. But a person who has blue eyes or a person who has brown eyes may have a blue eye gene within their system, but you don't see it. So the genotype looks at all those genes, recessive and dominant. So less than 1% of all prokaryotes, that's the bacteria, that's a single-celled bacteria, if you will, have been studied. Most don't grow in a lab. The new sequencing approaches revealing enormous biodiversity of the microbial world. In fact, 1,800 new bacteria species were found in Sargasso Sea alone. Major challenges remain. Exploring the microbial world should answer many of the fundamental biological questions. We're finding that by looking at the genotype, some of the bacteria we had in certain groups, under certain species, may not belong to those species, but they actually belong somewhere else, based on the genotype that they exhibit. These are the members of the microbial world. There's the microbial world. There's the acellular infectious agents, viruses, and prions. Here's the organisms that we talk about, bacteria, archaea, and eukarya. And it walks down the line and talks about all these different organisms that you see underneath those phyla, which is really the major separation. Now, members of the microbial world, there's enormous numbers. Bacterial species outnumber mammalian species by factors of 10,000. And they're present in all environments on Earth. Even the hottest thermal geysers in Yellowstone, and even in Antarctica, there are bacteria that grow there. And they're able to survive and multiply. The consideration of biodiversity typically overlook enormous contribution to microbes. And less than 1% of all micro species can actually be grown or studied in the laboratory. Kind of scary when you think about it. The outnumber is 10,000 to 1, and yet they're hard to study. Now, there are two basic cell structures we'll talk about. That includes prokaryotes and eukaryotes, which I referred to before. Let's talk about eukaryotes first. They're the more sophisticated of the group. They have membrane-bound nucleus, where their DNA is kept, and all these organelles. So the organelles are really small elements, small entities that do similar things that even our body has. For instance, nucleus is kind of brain. They have mitochondria, which produces energy. They have lysosomes, which work like digestion. They have other organisms, other parts of that sort of do the same things our organs of our body do. All of these things can be classified into one or three groups, like I said: bacteriae, archaea, or eukaryae. And organisms in each domain share certain important properties, and there are differences in them, obviously. And here you have bacteria, archaea, and eukarya. So prokaryotic cell structure, including the nucleoid, not a nucleus, a nucleoid, which is just where the DNA is kind of in. There's no membrane-bound organelles that separate them from the rest of the cell. Ribosomal RNA nucleotide sequences that are unique to this group. There are special RNA types, if you will. They're unicellular. The cell walls contain peptidoglycan, and the typical size is 0.3 to 2 nanometers. Now, archaea are prokaryotic cell structure. They include nucleoid. There's no membrane organelle, so it's similar to bacteria. The ribosomal RNA nucleotide sequence that are unique to this group, they're different. They're unicellular. They have no peptidoglycan, and their size is about the same, as you will, as is the bacteria. If you look at the eukaryotic cell structure, it includes the nucleus, and there's all the organelles or not all of them, but many of the organelles are membrane bound. Ribosomal RNA nucleotide sequence is different than these two, which helps us develop antibiotics that attack the ribosomes of these organisms. They're unicellular or multicellular. They have no peptidoglycan, and the typical size is 5 to 50 micrometers. Excuse me, nanometers. Sorry. So this looks at the size of the microbial world. There's a human there, and there's atoms here. We'll kind of walk through what that means. So there's adult roundworm or tapeworm. They can be seen. They're about this size. And as you go up with small molecules, which are atoms cooked together, we have lipids. This looks like a phospholipid that helps to make up the cells of our body. There's proteins. There's the ribosomes. We'll talk more about that later. They make protein in our body. There's the size of some viruses. Now, viruses have many different sizes. There's smallest bacteria. There's a prion fibril, if you will. There's a mitochondria, which is an organelle inside of a eukaryotic cell. There's a nucleus inside a eukaryotic cell. And most bacteria sort of fit in the middle here, and most eukaryotic cells. So you see that if you look at the unaided eye, you see basically you can see the roundworm of the human. If you want to look with a light microscope, you can see down to maybe a prion fibril if you're lucky, but everything else is going to require electron microscopy or something even more significant. Now, the size of the microbial world has enormous range. The largest eukaryotic cells are 1 million times larger than the smallest viruses, and the wide variations even within a group. Bacteria are about 600 nanometers and by 80 nanometers discovered in mid-1990s, visible to naked eye, and the bacterium is 70 times larger in volume, which was discovered in 1909. So eukaryotic cell is only one eukaryotic cell is as small as 1 nanometer, if you will, and it's similar to the size of a typical bacteria. But mostly, they're much larger, like I said. Now, scientific names use what's called a binomial system of nomenclature. And these are two words. One is like, think of the genus as the family name, like my last name is McGraw, and so genus is McGraw. The species is kind of like your first name. So in my case, it'd be Jerry. So my first name is Jerry, last name is McGraw. So genus is McGraw, and first name is Jerry. And so these organisms are named, their nomenclature is like that, last name and then first name. So last name of the genus is capitalized, and the species is not capitalized. Genus and species always are italicized or underlined, like E. coli, Escherichia coli. E is capitalized, coli is not. And they can be abbreviated with E period coli, if you will. And you can see the different techniques we use, the origin of the species to walk through for the scientific names. Now, to name bacteria are single-celled prokaryotes. The prokaryotes have a pre-nucleus, which means it's not a nucleus. There are no membrane-bound nuclei. There's no other membrane-bound organelles. The DNA is nucleoid, not a nucleus. Most have specific shapes, like rods or spheres or spirals. They have a rigid cell wall, which contains peptidoglycan, which is unique to bacteria, and they multiply through binary fission. 1 becomes 2, 2 becomes 4, and on and on. Many move using flagella. Not all of them, but many do. Archaea, like bacteria, archaea are prokaryotes. They're similar shape, size, and appearance to bacteria, but they multiply through binary fission. They may move via flagella, and they have rigid cell walls, but no peptidoglycan. However, like I said, no peptidoglycan. The ribosomal RNA sequences are different than the bacteria. And many are extremophiles, meaning they can live in high salt concentrations, high temperatures, low temperatures, that kind of thing. Eukaryotes have a true nucleus. That's what eukaryotes means. They have membrane-bound nucleus and other organelles. They're far more complex and sophisticated than prokaryotes. And the microbial members include fungi, algae, and protozoa. Algae and protozoa are also termed protists, if you will. Now, some multicellular parasites, including helminths, which are roundworms and tapeworms, are considered in this group as well. And this talks about all those groups, and you can take a look at that as you go through. Fungi, for instance, they're a diverse group. We have single-celled fungi like yeast, or multicellular fungi, which are like molds and mushrooms. A mushroom is really a fungus. Energy from degradation of organic materials. They literally break down organic materials. And they primarily live on land. There are a few that live in the water, but mostly on land. Algae, on the other hand, are a diverse group. They're single-cell or multicellular, and they are photosynthetic. They contain chloroplasts, which are organelles that contain chlorophyll, which translate light into energy. They primarily live in water. They have rigid cell walls, and many have flagella, like cell walls. Flagella are distinct from those of prokaryotes. They're a little different. Protozoa, they're a diverse group. They're single-celled. They're complex. They're larger than prokaryotes, and most ingest organic compounds. They have no rigid cell wall, and most of them are motile. They move around to get their nutrients. Now, parasitic helminths are worms that live at the expense of the host. They are not symbiotic. They break down the host. Adult stage can easily be seen without magnification, but the eggs and larvae are usually microscopic. Helminths include roundworms, tapeworms, and what are called flukes. And flukes are single-celled animals that can live in the lungs, lung flukes, and bladder flukes, and all sorts of things, different kind of flukes. Now, there are also acellular infectious agents like viruses, and prions, and acellular infectious agents are not technically alive. So viruses are technically alive. They're not microorganisms, so a general term as a microbe is often used to include them. So they're not microorganisms. They do not live. They don't have a visible metabolism. Viruses consist of either DNA or RNA. They're surrounded by a protein coat, and they're obligate intracellular agents, which means they live in cellular, that use the machinery and nutrients of the host to help to replicate themselves. So the virus will attack the host cell, enter it, reprogram the metabolism of the host, and to make more virus, which usually kills the host cell, and the virus escape and they move on. Or viruses escape and move on. Viroids, they consist only of RNA. There's no protein coat. They're obligate intracellular agents. They use the machinery and nutrients of host cells to replicate. And viroids are generally in plants. Prions consist only of protein. Believe it or not, they're just a chunk of protein with no DNA and RNA that's decipherable. They have misfolded versions of the normal cellular proteins that cause the normal versions they have abnormal versions that cause the normal versions to misfold. So they are just proteins with no nucleic acid, DNA or RNA included. Kind of makes them unique, and we're still not sure how they work. Now, viruses, nucleic acid packaged in a protein coat. There's a variety of shapes. They infect living cells. They turn the host. They multiply using the host machinery and nutrients, and inactive outside the host. In other words, they're obligate intracellular parasites. They cannot reproduce themselves unless they're using the mechanism of a given host cell. All forms can be affected by different types. So all forms of life can be affected by different types of viruses. Viroids are simpler than viruses. They require the host cell for replication, just like the viruses do. They consist of a single short piece of RNA. They have no protective protein coat, and they cause plant disease. So viroids are plant diseases, not humans. And some scientists speculate they may cause disease in humans, but no evidence has really been shown of that yet. Again, the scientific methodology has not proven that to be true. Prions, if you think about the mad cow disease that we had, the scare we had in the 1990s in Europe and in Canada, as a matter of fact, because they got much of their beef from Europe. Prions are infected proteins. They have misfolded versions of normal cellular proteins found in the brain. The misfolded versions force normal versions to misfold. In other words, it causes other proteins to break down. Abnormal protein binds these fibrils, and the cells are unable to function. Think of them as being put in a straitjacket. And they cause several neurodegenerative diseases in humans and animals. And they're resistant to standard cellularization procedures, so they're very difficult to deal with. Now, every rule has exceptions, so they're extremes in size. We have enormous prokaryotes, which are about the size of a tiny eukaryote. The smallest prokaryote, 400 nanometers, contains 1/10 as many DNA as E. coli. Internal structures, prokaryotic, like plantomyces, have membrane surrounding nucleus, nucleoid, if you will, and they carry out endocytosis, which means they have nutrients which come into the cell, are worked on by the cell, and then are removed from the cell through that process. So they practice endocytosis. So even those have an exception. So now we've talked. This has really been the introduction of microbiology, the first chapter. Thank you very much.